All right. Hi, everyone. As a UC Santa Cruz Alumni Council member, I'd like to welcome you to our monthly speaker series, Slugs and Steins, Lectures from UC Santa Cruz. I'm April Yee. I graduated from Oaks in 2002. And for those who are new, our Slugs and Steins series engages a UC Santa Cruz faculty member in discussion with you, the local community of Silicon Valley, and our extended community online, with a goal of making us all Renaissance people. We want it to feel just like you're at UC Santa Cruz sitting in class, but with drinks and without tests. Mike Reapy, another volunteer organizer, is with me tonight. We're both alumni. I'm a holistic life coach focusing on transformative self-care for professionals, and Mike spends his days as an engineer, Silicon Valley style. And he'll be helping me with the Q&A, and you'll hear more from him at the end. So before we get started, and since we can't see you, we'd like to know where you're zooming in from and how many people are watching. So please take a moment to fill out a short poll that will pop up on your screen. And we'll share the results after we've given everyone a moment to respond. All right, it looks like most of everyone responded, and you should be able to see the results of our poll and who is in the virtual room with you. So mostly just single folks watching, some pairs. No one responded from outer space today, which is rare. Someone usually does that. Lots of folks from Santa Cruz and the Bay Area. One person from outside the US. Very cool. So tonight, we will be tipping our Steins to this evening with Professor Carrie Partch. Professor Partch earned her Bachelor of Science degree in biochemistry with a minor in Italian from the University of Washington in 1997. And she conducted research at the Oregon Health Sciences University in Portland, Oregon, before attending graduate school at the University of North Carolina in Chapel Hill. She worked with Nobel laureate Aziz Sankar to study circadian rhythms and the blue light sensor Cryptochrome in bacteria, plants, and animals, earning her PhD in biochemistry and biophysics in 2006. She then trained in structural biology techniques with Kevin Gardner and Joe Takahashi at UT Southwestern Medical Center. Since 2011, her lab at UC Santa Cruz has focused on dissecting the molecular steps that generate circadian rhythms from humans to bacteria. Her group has been recognized with several awards, including the 2016 Junior Faculty Award from the Society for Research on Biological Rhythms, the 2018 Margaret Oakley Dayhoff Award from the Biophysical Society, and most recently, the 2002 Award in Molecular Biology from the US National Academy of Sciences. Additionally, one of Professor Partch's students, Alfred Manning Freeberg, who she mentored, won a Dean's and Chancellor's Undergraduate Research Award in 2020 for his research in chemistry, his chemistry senior thesis project titled Anion-Driven Conformational Changes in CK1 Delta Regulate Circadian Rhythms. So we'll address questions at the end of the talk, but you don't have to wait until the last minute. You can type your questions in at any time. And if you see someone else's question that you like, you can also upvote it in the Q&A box and we'll ask it sooner. We'll prioritize it. And this talk is being recorded. So in a few days, you'll be able to find it on the UC Santa Cruz Arts and Lectures YouTube channel. We'll post the link in our social media channels and follow-up emails. Okay, so does everyone have their Steins? I have my tea. Well, I've got your slug, Professor Carrie Partch.
Thank you so much, April, for that kind intro. It's such a pleasure to be here today. to tell you about our work focusing on biological rhythms that align our lives with the daily rhythms of the Earth. So I'll start off by giving kind of a brief overview of why we think circadian rhythms are so important and pervasive amongst life on Earth. And so, as we're seeing here on the left, just to remind you that we live in an ever-changing environment, that as life evolved on Earth, there was a constant cycle of light and dark, of hot and cold, and life evolved to this ecological niche by building biological clocks that allowed organisms to keep track of 24-hour time within themselves. So here I'm showing you a handful of photosynthetic organisms from the beautiful single-celled cyanobacteria, Synchrotron elongatus, all the way to sunflowers, each of which, of course, has a really important relationship with sunlight. It's food. And you can imagine during this daily cycle of light and dark, it's really important to keep track of the sunlight, because when it becomes nighttime, food is gone, and without a biological clock to know that the sun is going to rise in the morning, you may well spend all of your energy resources before you see food again. So I'm going to show you a video that I think really beautifully captures the power of circadian rhythms. So here we're looking at a baby sunflower, and it's kind of woken up for the day, and it's tracking sunlight or food by moving along with the sun as it moves across the horizon. And here, as the sun goes down, for lack of a better word, the plant goes to sleep. But here's where you see the power of the clock, that it wakes up before dawn and it points east to catch those first few delicious photons of light that come up in the morning. So this is really why we think that clocks are so important in physiology, that they allow us to predict regular environmental changes that occur throughout the day. It turns out it's not just photosynthetic organisms that have clocks that keep track of the day, but most forms of life on Earth, all the way up to humans. And these clocks you can think of as being one of the most basic and fundamental adaptations to being a form of life on Earth. And so I don't know if any of you have ever seen this crazy subway diagram. This is a map of your metabolism. And every single one of these dots is a gene in your body that helps break down food that you eat and get those nutrients to the right place in your body at the right time. Now imagine you've just had a big bolus of sugar. That sugar will take a ride on the subway down the metabolic train to this so-called TCA cycle, where your body will produce energy in the form of a molecule known as adenosine triphosphate, or ATP. So this is a regular subway station stop for sugar in your body that helps give you energy. Now, much like a subway, they don't run the same at night as they do in the day. There's reduced service during the evening when there's reduced demand. So one thing I hope to get across to you is, even though plants use their clocks to align their metabolism with the day-night cycle, because they have to, because sun is their source of food, we do the same thing too. So if you undergo circadian disruption, this can happen as a consequence of shift work or jet lag, travel, transmeridian travel. You basically disrupt circadian rhythms within your body. And we now know that when you do that, all of the genes that are indicated here in red are misexpressed and miscontrolled in your body. So in other words, when your circadian rhythms are disrupted, you too have issues with your metabolism and how you process food. And I think this is really beautifully summed up by work from Sachin Panda that was done about a decade ago, where they fed mice either a normal diet throughout the day or ad libitum, or they restricted the diet to the evening. Now remember, mice are nocturnal, so this is the equivalent of them eating during the daytime for us. So they eat when they're active. They also fed them what's known as the high-fat diet, which sadly is also called the Western diet, and they did the same time series. So you can see here in this graph that no matter what the diet was, all of these mice ate the same amount of calories every day. Mice aren't stress eaters. They don't overeat. They eat until they're calorically satisfied and they stop. But what's important here is that this study showed that when you eat may be just as important as what you eat, because the mice that were on the high-fat diet exhibited dramatically different weight gain depending on when they ate. So mice that restricted their eating to the nighttime when they were active only gained about 10% more weight than the mice on a normal diet. But the mice that kind of nibbled around the clock became morbidly obese. So hopefully I've been able, just in this very quick intro, to explain to you that clocks are important because they help root us in our day-night life that we have on Earth. And things like as fundamental as metabolism, what happens to the food we eat, is under circadian control. But to me, it's really remarkable. So I want to walk you through a few of the other things that are controlled by the clock, just to set the stage for the rest of my talk. So, one thing that you most probably immediately think of when you think of circadian rhythms is the sleep-wake cycle, which is under control of the clock. So somewhere, a few hours before a typical wake up is your lowest body temperature, and then it sharply rises to help prepare your body for the time to wake up. Now, right after you wake up, we know that the sleep-wake cycle is one factor that controls it is the release of the hormone melatonin. So this is low during the day, and then it increases steeply as the sun goes down at night, so that it promotes the feeling of sleepiness to make you want to go to bed. So right after you wake up, you have a sort of an increase or jump in your blood pressure. And actually this circadian-driven increase in blood pressure in the morning is linked to a 2 to 3-fold increase in heart attacks and strokes in the morning, because the body sort of jams up your blood pressure to help prepare you to be alert during the morning. Now, some of this alertness is good. We do know that the time of day where you have the best ability to focus and concentrate is kind of late to mid-morning. So I often joke when I present to school children that they better schedule the hardest class in the morning before lunch, because it's statistically proven they'll do best. Now, many of us after lunch kind of feel a dip in energy, and it's now been proven scientifically that the best time for a cup of coffee to sort of awaken your body and get you through the rest of the day is right after lunch. Now, there's lots of fun things that we can talk about as well. For those of you who are fitness buffs, you have the best ability to add muscle mass when you exercise late in the day. You sort of feel the least pain and you have the best ability to add muscle mass. So rather than an early morning run, you may want to think about a late afternoon run. And of course, all of these things that seem really broad physiological outputs of the clock also are involved with some of the more deepest, most core molecular events in our body. For example, your ability to repair damaged DNA in your body also undergoes circadian rhythms, with a peak just at the end of a long day in the sun. So really, I'd like you to think about circadian rhythms as controlling many different aspects of the timing of your daily lives and integrating them so you're active, hungry, and alert during the day, and restful sleep and not eating at night. Now, I'm going to walk you through the molecular basis for circadian rhythms. This is what we work on in my lab, so that we can understand the way that we keep time at the molecular level and how this is affected by people who have sleep disorders. So there are two genes, cleverly known as clock and BMEL1, that work together by binding DNA at various sites in your body. We can think of these as the activators or the gas pedal of the clock. These particular proteins help to transcribe other clock genes known as period or cryptochrome, cry for short. When these proteins accumulate, they interact together in big complexes, and they come back in and turn off the activity of clock and BMEL. So we think of them as the repressors or the brakes of the circadian clock. Now, looking at this network, it may not seem intuitive as to how timekeeping arises from this loop. But I'll show you an example, and actually several, that I think will illustrate this really nicely. So you can think of any clock control protein that at dawn, the levels of that protein are low, and that as that protein is made by clock and BMEL, it then gets shut off at the end of the night by the brakes or the repressors of the clock. So you get activation in the morning, turning it off during the night, and you get this beautiful rhythm that goes up and down once a day. And in fact, the time it takes to get from one peak of clock protein expression to the next is what sets what we call the period or the timing of the clock. Now in humans, this is just about 24 hours. And every day this synchronized via light that you see through your eyes so that you line up exactly to the 24-hour light environment wherever you are. Now it's not just the 4 genes that I've indicated here that undergo this beautiful oscillation up and down in gene expression, but nearly 10,000 genes in your body do this every single day. So something like over 40% of your genes are kind of humming along, being made at specific times of the day to control when your body, in fact, is going to repair DNA or add muscle mass or break down that sugar that you eat. So really it's a network of this beautiful change in gene expression. Now, within our bodies, we have a hierarchical system. Now, you may have heard of the so-called master clock in our body or our brain that gets light information. This is located in the hypothalamus, right behind our eyes. In fact, it's the first pit stop that light makes when we perceive it. First, it goes to the SCN to tell our bodies what time of day it is outside. Then it goes to our mood centers in our brain. Then it goes to vision, just to give you some idea of maybe where the brain thinks things are important. Now, this SCN clock is the one that is working in lockstep with the light environment. And it communicates this information to clocks in the rest of your body through changes in metabolites, hormones, and even changes in body temperature throughout the day, so that clocks present in essentially every cell in your body are keeping time and walking in lockstep with the master clock. Now we can look at this through some advent of biotechnology. So we can take the luciferase gene from fireflies that allows it to glow, and we can basically put it in the DNA right behind the period protein so that we can watch this clock tick. So here you're seeing a slice of the SCN from a mouse tissue, beautifully oscillating, giving rise to this undulating pattern of gene expression that's keeping time and telling the rest of the body kind of what's going on in the outside world. Now, hopefully you can see that these are kind of beautiful, complicated structures. Oops, trying to get my mouse here. Each of these lobes has around 10,000 cells within it. And for a long time, it actually wasn't known whether or not it was this entire brain structure that was important for the clock, or whether the clocks happened individually within single cells. And so I'll tell you the answer is that within every cell of your body, you have a clock. But here in the SCN, they group together to keep time and really keep everybody working at the same time. So here's a picture of individual mouse SCN neurons. Now the resolution is a bit grainy here because each of these specks of light is a single neuron. It's just marching on it to its own beat and keeping its own time. In fact, the cell that's highlighted here in the box, we're looking at changes in the luciferase or light intensity down below. And hopefully you can see that there are beautiful circadian rhythms, even at the level of a single neuron. So it's this network of clocks in your body that work together to help synchronize the activity of all of your organs and tissues with your brain and keep them aligned to the light-dark cycle. So about 20 years ago, Chuck Seisler at Harvard did a really extensive study and showed that across the human population that on average, the time it takes humans to go one time around the feedback loop that we've shown on the left is just over a day. So 24 hours, 11 minutes, plus or minus a few. And that this really is the timekeeping. If we were to put you in constant darkness and you did not receive light cues, this would be the time that your body clocks kept. So I really want you to think of these clock proteins as cogs in a molecular clock that keeps time, where they work together with one another to give rise to this very specific timekeeping. And our lab works to try and understand how these proteins interact with one another to give rise to 24-hour timekeeping. Now, there's a really amazing questionnaire that's floating around called the Munich chronotype questionnaire. And this is something, a chronotype is what clock type are you? What preference do you have for the daytime? If you have a clock that's pretty much average, around 24 hours, 11 minutes, then you're going to kind of fall right into the middle of the spectrum. So we're looking at sort of the percentage of the population here that perhaps go to bed at like 10 p.m. at night and prefer to wake up around 6 or 7. So I know that Kristen had a poll that we were going to send to ask you what your chronotypes are and how you identify. A morning lark is someone who likes to go to bed earlier than say 10 or 11 and is much happier getting up before 7 a.m. A night owl, of course, is someone who prefers to go to bed at midnight or later. And then that kind of middle of the road is someone who prefers a bedtime around 10 p.m. So it's interesting to see the poll results come in because we kind of have the night owls are like winning here with a little more than the rest of the population. And this actually is pretty typical. So I'm gonna end the poll now, so I can move on with the talk. But we're going to talk about the molecular basis of this for the rest of the talk and the changes that we've been identifying and studying and how these changes give rise to our behavior. Okay. Let's see, let me just get this out of the way now. Okay, perfect. So as you may notice, there's a long tail here in the late chronotype. So there's more people who tend to skew late night than morning folks. Sorry, it's not letting me move on with the slides. Okay, so I want to illustrate that morning larks are sort of, the early chronotypes, and night owls are the later folks. And we now know that this behavior actually changes throughout age. I just did this Munich chronotype questionnaire, and I found myself to be a so-called moderate morning person. I'm one of the lucky 20% of folks who don't have to wake up with an alarm clock. Okay, so we know that chronotype can change with age. That often when we're about 10 or younger, we have very early chronotypes. We go to bed early, and parents of young children will know they typically wake up quite early. Now something really extraordinary happens as we enter our teenage years. Our chronotype goes from being very early to very late. So for example, I have a 17-year-old and he routinely stays up well past midnight and likes to get up quite late. So as a circadian biologist, the mother in me wants him to get sleep, but also knows that he's following his biology. And in fact, a lot of school systems have recently moved to push back the start time of high schools because kids in their late teens are almost at their peak late chronotype. And beautiful studies out of the Seattle public school system have shown that pushing the clock back just 1 hour led to a several percent increase in grades across the board because students were more well rested and better able to pay attention in the morning. Now, something interesting happens here in our mid-20s for reasons that we don't really know yet at the molecular level. We begin what I call the long slide to the early bird special. So our chronotypes get earlier and earlier for the rest of our lives. And one interesting thing about Till Roneberg, the researcher who published this paper, is that he actually suggested here that men might like to date younger women because they have the same type of clock or chronotype. So you can think about that tonight and see how that sits with you. Okay, so in all seriousness, back to chronotype. So, we know that there are many factors that lead to changes in how we like to be in our clock type or chronotype. Sometimes this is due to altered sensitivity to light. So light is an incredibly important stimulus for our clocks. During the morning, light is an essential cue that tells our body that it's time to get up, it's time to be active. And I would really encourage all of you to make sure to get out in the sun, the bright outside daylight for at least 30 minutes every morning. It's a hugely important cue for your circadian rhythms. Likewise, light at night is bad because it tells our body that it's daytime. So some folks have altered sensitivity, which influences their ability to line up with the day-night cycle. But one thing that's really interesting to our lab is that we now know that there are inherited changes to the so-called clock genes that make up circadian timing that give rise to changes in the timing that this clock keeps. And so as a scientist, these mutations are incredibly interesting because some of them make the clock shorter and some make the clock longer. And those tend to skew towards morning lark and night owl, as you see here. And when I say shorter, I mean, there are some inherited changes to the genome that people have that make their clocks 4 hours shorter than normal. I mean, it's incredible. So they can never really catch up to the day. They're living kind of constant jet lag. So I'm going to denote these types of changes that cause early chronotypes in purple and late chronotypes in red. So going back to our map or model of the clock, you can see here that we've actually found many different mutations in animals and humans that give rise to short clocks that make these critters and people morning larks. And really our first clue came from this little guy. This is a Syrian hamster. So he, along with Michael Menaker, who's shown here studying them, Michael would watch and monitor as these animals would run in their wheels. And so these particular creatures are nocturnal. So here you can see a record with every day is one horizontal line. And when they're awake at night as nocturnal animals, you can see them kind of these thick purple bars are showing you when they're running in their wheels. So they're awake at night, they're running, then they kind of go to bed. Now, hopefully you can tell that when you plot this on a 24-hour scale, these hamsters have a near perfect precision where almost every day, in the absence of any kind of light cue, they wake up and start running at the same exact time of day. So they were a really fantastic model system back in the 1970s and 80s when Michael Menaker was first trying to understand the molecular basis for human circadian rhythms by studying this little nocturnal hamster. Now, there's some incredible stories about this that maybe I can tell in the Q&A, but out of luck, his graduate student, Martin Ralph, found one hamster that just behaved differently. When he got up, and came to the lab to study these hamsters, there was one hamster that was always up and running before the others. And when they bred that hamster, they found out that in fact, that change was heritable. And they named that particular mutant the tau mutant for time or period. And hopefully you can see this is still plotted on a 24-hour scale, but you can see that every day it starts 4 hours earlier than the next. So that rather than lining up beautifully in a 24-hour environment, there are times in the day, a 24-hour day when they may be asleep or awake compared to the normal mouse. So this is a mouse that just cannot live in a 24-hour world. Now, it took about a decade, but Joe Takahashi, my postdoc advisor, identified it as being a mutation in this key kinase, casein kinase 1 delta, or CK1 delta for short. Now, if you fast forward another 20 years, our lab showed that just change of one amino acid in that kinase led to a dramatic change in the structure of that kinase. So here I'm showing you just an overlay of two very high resolution crystal structures of the kinase, the normal protein in blue and the tau mutant in gray. And you can kind of watch the protein as it flips back and forth. And it's this little tiny change in the kinase that supercharges it to do its job, which is to promote the clearance or turnover of these clock proteins. And in their absence, that clock that we're talking about starts sooner. So it's just one change in clearing out this repressive complex helps shorten the clock by up to 4 hours. So how is it that we go about getting this kind of data? It's kind of molecular insights that allows us to understand the details of the molecular clock and how it can go awry if you inherit a mutation, say from your mom or your dad. So the way that we do that typically is through X-ray crystallography. So these are crystals. You may be used to looking at salt crystals, which are quite large. These crystals for scale here are a purified protein, and the scale bar is about the width of a human hair. So they're incredibly small crystals of highly purified protein. They're so small, in fact, that we have to work, our students have to work using microscopes. So here are two students actually looking at a crystal plate that's just out of view here. And hopefully you can see in this picture here's a drop of liquid here that has a bunch of really beautiful shiny crystals in it. So just for scale, this drop of liquid that I'm showing you is about 1/1,000,000th of a liter, or 1 microliter. So we make very tiny drops out of precious protein to get them to form crystals. And then using a molecular kind of lasso. So here's a tip, and hopefully you can see one crystal here that has this incredibly thin nylon string. So we lasso these crystals out of this tiny drop of liquid, and we quick freeze them in liquid nitrogen, and we take them to the synchrotron. So the synchrotron is a particle accelerator that gives us the high energy X-rays for which we shoot at our crystals to give us hints of the protein structures and shapes within them. So this is a photo that I snapped with my lab when we went to the advanced light source at the LBNL lab just up in Berkeley. And I want to show you a movie that I collected from one of my students, Nikki here, who was an undergraduate with me several years ago. She went on to Stanford to get her PhD and actually just graduated. So here you can see Nikki pulling out her frozen crystal using this magnetic loop, transferring it here to some forceps. So we'll keep it bathed in liquid nitrogen so it can stay frozen. And she's going to transfer it to the beam line very carefully without dropping it. So here she's putting on the beam, and the crystal is located at the very end of that tiny, tiny pin. She's going to close the door, turn the photons on, and we're going to hit that sucker with some X-rays, right? And so it's just a really fun hands-on exercise. So here's the picture of the actual loop with magnification that Nikki loaded. And hopefully you see that kind of lasso with this big rod-like crystal. And this circle is exactly where the X-ray beam is going to hit. Now, when we, to every scientist's glee and surprise, when we hit that guy with X-rays, we saw this beautiful pattern of diffraction spots that told us there were protein molecules deep within that crystal that would give us the secrets of their atomic structure. So we do, in fact, use a pattern of diffraction spots like you see here that we collect at the synchrotron. And we work backwards with the help of many different computer algorithms to build a mesh, if you will, of the molecular structure within that crystal that gave rise to those spots. And this is how we solve structures of proteins like what I'm showing you here. So this is a high resolution crystal structure of the clock BML1 dimer bound to its cognate sequence of DNA down below here in gray. And it's structures like this that really help us understand how these proteins do their jobs to tell time. So here's another structure of a clock protein called Cryptochrome. And I've painted this big, deep pocket, this cave-like pocket here in green, because work in my lab several years ago showed that this pocket was important for Cry and the rest of the repressor clock proteins to bind onto clock BML and sort of initiate the repressive phase of the clock. So I'll just give you a little bit of a tour. Like people, proteins come in all shapes and sizes. And we knew for a long time that these two proteins interacted. And in fact, it was this knob on the end of the structure that fits into this pocket, much like a Lego, that helps us understand how these two proteins come together at dusk to begin the phase when clock gene expression turns off. So I'm going to take you back to this model of the clock. It actually was a recent discovery of a late night night owl allele in Cry1 that caught our attention because so far it's the only known human inherited change in our genome that makes us night owls. And so, I'm not a trained sleep biologist, but I'll show you kind of how these were measured. And, hopefully you'll come with me and realize that, you don't need to be a sleep biologist either. So what we're doing is looking at, unlike the hamster record before, we didn't make people run in wheels. We actually monitored their activity with something like a really fancy Fitbit called an actigraph. And when you're active during the day, you give rise to these black tick marks. Now the red triangles indicate when the subject has elected to gone into bed, And for most people, they go into bed right around midnight, which is shown on this line. So almost like clockwork, I guess pun intended, these folks go into bed and they come out of bed, just a little bit after 8 a.m. almost every day. So a beautiful, regular, routine sleep record. Now, this is a bit different with folks who inherit this night owl allele known as Cry1 delta 11. It does not take a trained sleep biologist to notice that they're often going to bed in the middle of the night. So this time it's 4 a.m. They didn't go to bed until 8 a.m. here. They're almost never getting 8 hours of sleep. And you can see that the beautiful record above, which represents a really good sleep hygiene of going to bed and getting a good night's sleep and waking up, is really obliterated in this Cry1 delta 11 subject. And so, we were incredibly interested in finding out the molecular basis of this night owl change, and if we could try and fix this perhaps with therapeutic approaches. So here's the title of the paper that showed it, that this mutation in Cry1 gives rise to familial delayed sleep phase disorder. This delayed sleep phase disorder is the fancy scientific term for night owl. And what I want to do is kind of show you the change in the genome. So really, each of these numbered boxes is a so-called exon, or a piece of DNA that encodes for the Cry protein. And they all get kind of chopped up and put together. Now, one single change from an A to a C is all that's needed to do that to your sleep cycle. And in fact, what happens is that single change in the Cry1 gene causes exon 11 to be skipped or spliced out of the protein. So when your body makes the Cry1, it simply skips over this piece of the protein and omits it from the balance. And so this really ties in to the most fundamental basis of my own scientific career. So as a graduate student, 2 decades ago with Aziz Sundar, I was very interested in studying cryptochromes, which had just been discovered as key regulators of the mammalian circadian clock. Now, cryptochromes were interesting to our lab because they were evolutionarily related to a really ancient DNA repair enzyme known as photolase, which is shown here on the right. So photolases were really the initial scientific discovery of my advisor, Aziz Sundar. And he was the first to clone this gene and show how it uses blue light to repair UV-induced damage in your DNA. And for this, he was one of several recipients of the Nobel Prize in Chemistry in 2015. So when I started in Aziz's lab in the year 2000, all we knew was that these two proteins were related. And now with the benefit of structure, we now know that this beautiful cavity, which is also present but much smaller in photolase, where it uses it to bind a light harvesting cofactor responsible for repairing DNA, that this same pocket is incredibly important for Cry function because it brings cryptochrome together with its clock protein partners. Now, as a graduate student trying to figure out 20 years ago without these structures what cryptochromes did, one of the contributions I made was in fact to see that there were some changes in what we call the photolase homology region, or PHR. So these two domains have similar overall structure. But I also discovered that cryptochromes have kind of an enigmatic tail that doesn't have a beautiful structure like we see here, but in fact is disordered. So you can think about this as kind of a wet spaghetti noodle. It's thrashing around, very dynamic. And because photolases did not have this tail, we guessed that it must be important for cryptochrome function. But back in 2005, we didn't know what they did or how they worked, just that they existed. So if you fast forward 15 years to Mike Young's discovery, the deletion of exon 11 in this tail causes dramatic changes to human circadian timing that really have a profound effect on our day-night sleep cycle. I was elated to see that this wiggly tail may have some important role in cryptochrome function. So I'll remind you that cryptochrome and clock and beam all bind, And they interact through this, pretty well-studied pocket, knob and hole pocket interaction. So really, when thinking about how exon 11 may be playing a function, in the beginning, we really had no idea. We thought somehow exon 11 must be interfering with this interaction in some way, such that in its absence, when it's not there with folks who inherit this NAGL gene, that cryptochrome combines to clock and BMO more tightly. So this was shown in the original paper, that somehow the interaction of these two proteins was turbocharged in this NAGL mutant. Because these proteins stay together longer, it drags out that molecular clock, in fact, making the period of the clock longer. And in doing so, it delays when melatonin is made and released in your body. So it's about a 30-minute change in the period in your body, but it delays the release of melatonin for up to 3 hours. That's what ultimately makes it such that folks who don't have exon 11 in their Cry1, what makes them go to bed around 2 or 3 in the morning. So my student, John Carlo Perico, working with an undergraduate, Yvette Perez, decided to ask a series of questions to explore how exon 11 works to control circadian timing. So to do so, we broke the protein up. So we took this isolated disordered tail and we studied it by NMR spectroscopy. Now you can basically think of this as doing an MRI on an individual protein. So we put these proteins in a test tube, we put them in a giant magnet, and every single one of these dots that you see corresponds to one amino acid in the tail. And it tells us about the environment of that tail so that we can ask questions like, does that Cry1 tail bind to its own PHR domain? Do they make an interaction with each other? So to do that, we took this isolated tail and we added increasing amounts of the protein to it to see if in fact they made an interaction together. And what we saw was that some amino acids, like the one I'm showing here, exhibited no change when we added the PHR domain in, indicating that it was largely free of the PHR and moving around, while others lost their signal intensity, telling us that they were in fact making a direct interaction. So this is a really beautiful and unbiased way for us to map, kind of without putting our initial presuppositions on it, to map how the PHR domain might interact with exon 11. And in fact, we were able to map all of these different amino acids that interact with the PHR to exon 11 and some that were right next to the PHR domain. So this is really beautiful evidence that helped to tell us that in fact, exon 11 does in fact bind somewhere to the PHR domain to control its function. So the last thing that we did that I'll tell you about is that we went the next step to ask, okay, if they bind, does exon 11 affect the ability of Cry1 to bind to its partner? And this was really something that was expected given the data that was published in the initial report. So we have ways to very quantitatively measure how tightly two proteins interact. And so if we take the full-length protein that has the tail, it binds with modest affinity. So the higher this number is, which is the dissociation constant, the weaker these proteins bind. And so here we can see that the full-length protein binds with modest affinity, but if we chop this tail off entirely so that we only have the PHR domain, we actually bind much more tightly. So the real test, the really exciting day in the lab was when Carlo and Yvette made a version of the protein that was just like those that you inherit as a NAGL, that was lacking exon 11. So it had the tail, just not the 24 amino acids encoded by this particular piece of DNA. And in fact, that protein interacted just as tightly as if the tail wasn't there. So this, along with other evidence, was really conclusive that exon 11 works to compete with clock and regulate how tightly these two proteins bind. Now I have the tried and true Family Guy cartoon way to explain this science. You can think of Cry1 as this ball and cup game, where the PHR domain is the cup, that the ball is being tethered by this flexible disordered linker, and in fact the ball is exon 11. And in this cartoon, you can see that it's bound in many different ways. It's up, down, around. Sometimes it's bound to the PHR domain, as shown here on the left. And this really illustrates what we think of as the dynamic nature of this tail. Now, at the same time, we know the clock and BML really want to bind to the same site, this cup. And so the idea fundamentally is that when exon 11 is bound to the PHR domain near this pocket, the clock and BML can't. And it's this really a subtle fine-tuned balance of protein interactions that wreaks havoc on the molecular clock of people who inherit this particular mutation. So hopefully a Family Guy was a decent way to explain some molecular biochemistry. So the question is, can we use our knowledge of night owls to help people get a good night of sleep? And so right now we've partnered with a company called Atomwise to start looking for drugs that can bind within this pocket and take the place of this, the job, if you will, of exon 11. And in doing so, we help to restore this kind of really disturbed, altered sleep pattern that's incredibly prevalent. So 1 in 75 people of European descent have one of these alleles skewing their sleep to a very late pattern. This is incredible. Millions of people on Earth have this issue of really not being able to get a good night of sleep before work. So the question is, if we find small molecules that bind into that pocket, if we find how to therapeutically treat folks, can we restore a good night of sleep and all the kind of benefits in life that come with that? And that's really one of the next stages of our research to try and do that. So I've kind of framed all of this as kind of cute tale about when you like to go to bed and when you like to wake up. Although I will admit, if I was an extreme night owl like that, it would be hard to get up and go to work in the morning. But I really want to end by emphasizing that circadian rhythms align fundamentally all of your behavior and physiology with the light dark cycle. Everything from when you feel hungry and what you do with that food, to how you feel and think throughout the day, to basically everything that's fundamental to your physiology. How you make hormones, when you make them, how your heart and vasculature function throughout the day, to even when cells divide in your body. So this is, the last point I'm going to end on is that we know that there are links between the cell cycle and rapidly dividing tissues and circadian clock. The clock essentially gives permission for cells to divide once a day. If you look at this map of the Earth, that was a beautiful picture that was constructed by NASA, you can see the two faces of Earth, the light side and the dark side. But hopefully you notice here the really pervasive influence of light at night. And this is what we think, as circadian biologists, we think of the rise of light at night with industrialization as one of the key factors that's disrupting circadian rhythms and all of the sort of healthful rhythms that go along with it. And so we know that, for example, shift work has been declared by the World Health Organization as a probable carcinogen because it alters the clock that fundamentally takes your cell cycle rhythm out of this beautiful alignment with the day-night cycle and can increase the risk of breast cancer by up to 20% for long-term shift work. And so we know that we can harness the power of the clock, that there are really exciting days forward. It's not all doom and gloom about light at night, but understanding fundamentally the rhythms of our clocks and how they integrate and control all of our physiology is really opening a new day on understanding how the drugs that we take interface with the proteins in our body. And in fact, there are almost 1,000 FDA approved drugs that target genes that are under clock control. And there's a kind of new wave of science really trying to understand if we can take drugs at the right time of day when they have their best efficacy, we may be able to either lower the doses that we take or take different amounts that would have increased potency, say, to fight cancer. So I don't know if any of you caught this, It's really fortuitous timing. There's an incredible piece in the New York Times magazine that just came out this weekend called The Quest by Circadian Medicine to make the most of our body clocks. And if any of you have found circadian rhythms at all interesting, I would really suggest that you take a look at this article to really round out your understanding of how clocks control our body, our health, and what we can do to make them work better. So with that, I want to thank, this is everyone who's been in my lab over the last decade at UC Santa Cruz. And in particular, it's such a joy to highlight this, speaking to UC Santa Cruz alumni, because these folks are the people who have done the work that you've seen, who have helped to build new and fundamental understanding into circadian rhythms. And without them, I really don't think we'd have kind of the molecular picture that we do. So I will thank my funding. So we had a lot of funding from NIH, in particular the National Institute of General Medical Sciences, the so-called Basic Science Institute. We've had a lot of support from national labs where we collect our synchrotron data. We've even had support from the Santa Cruz Cancer Benefit Group when we studied some clock cancer overlap. And finally, I just want to say a huge thanks to Yuli Ortega for running the UC Santa Cruz STEM diversity programs. This is just an incredible suite of programs that are there to help first generation students and students from underrepresented and minoritized backgrounds get into science and really bust open a whole new career. I support the UC Santa Cruz STEM Dreamers every year on Giving Day, and I've included a link down here if you're interested in helping to support young students get into labs and have meaningful research experiences that put them on a path to becoming world-class scientists. So with that, I will stop my talk, and I would love to take any questions. Thank you so much.
And thank you so much, Carrie. Again, my name is Mike Reapy. I'll talk to you again later, but I want to thank everybody for your questions. We have about 16 questions in the Q&A box. I'll just remind everybody, if you have some questions for Professor Partch, please type them in the Q&A box now. You can also take a look through other people's questions. You can upvote them and we'll ask them sooner. So I'd like to start with a question from Tobe. Any thoughts on daylight savings time? Maybe I'll say data on daylight savings time, right? As Congress looks to maybe kill it, I've heard that it's very important which way they kill it. Should it be the fallback or standard time, or should it be the spring forward daylight time slot? I've heard that picking the wrong option could have a significant health impact, such as increase in cancer and likely heart attack or stroke.
Absolutely right. So we know that standard time is basically Earth time. Standard time is such that at noon the sun is overhead or should be close to overhead, and our bodies, have evolved for thousands of years, right, to adjust that. So it really is worthwhile to limit this daylight savings, but to keep it back in standard time and not the advanced time that we're in right now, for example. So to get to your point, we know that there are data showing that across a time zone from the easternmost edge, which is kind of the earliest, to the westernmost edge, which is the latter end of that time zone, there's an incredible change in cancer incidence. That on the West Coast edge, which represents kind of like the latest, most daylight savings time, the cancer incidence spikes. And there's a ton of information that not just the flipping back and forth is quite bad. Anecdotally, it takes me several days to really feel caught up. I don't know about you, Mike, but I don't feel right for a few days. But we know that car accidents spike. There are accidents, many other types of accidents spike when we flip our clocks back and forth because we're asking our bodies to adjust an hour kind of arbitrarily. So, the science is really clear that sticking with standard times is the best for our bodies. And I know that there are some folks like JP at stevestandardtime.com who really have a ton of information if folks are interested in reading more of the science-based evidence for that.
Thanks, Carrie. Wow. I mean, that I would appreciate having standard time as our standard time around the during the year. And I know I would have also loved to have started high school a lot later than 8 a.m. every day. Sometimes we started at 7 a.m., which was so crazy.
Yeah, I think just in California now, I read the other day that starting next year, no senior high school can start before 8 or 8:30. And so, there really is a ton of scientific evidence that moving school time back just a bit really helps children get a good night of sleep and perform better at school.
Yeah, thank you. Here's a question from Rick. I have seen studies of NBA games that suggest 4 to 7 local time is not peak performance. Are you aware of such studies, or is that not supported by the evidence?
That's intriguing. I haven't seen that particular NBA study, but I've seen that like one thing that there are two things that work together to suggest that it might be best to work out in the late afternoon. One is that your pain threshold is at its highest. And the second is that you're most able to add muscle mass. So if you're really pushing it, if you're a true CrossFit aficionado or something, if you're really pushing it, then the end of the day is supposed to be sort of the best time to do that.
I noticed Rick said local time. That might also be a question about, traveling for games, maybe shifts your clock, right?
Oh, incredibly. Yeah. I mean, if you think about it, we, up until the last, 50, 60 years, we did not have the capability to outrun the sun in a day. We travel by car or horse or, whatever, mule, what have you. And so really it's just the advent of sort of travel, business travel, flights that allowed us to really explore the consequences of jet lag, which are just profound.
So there's a question from John. There's a supposed connection between individuals who first suffer depression and altered sleep patterns. Is there any work that connects clock proteins with depressive symptoms?
So there's really beautiful work from Colleen McCline and others kind of exploring both ways, how alterations in clock genes can lead to like manic like behavior or depressive like behavior. Again, most of the studies I'm talking about are done in like mouse model systems where we can explicitly say disrupt the clock. But we know from human studies that for those folks who have, say, like even bipolar disorder, that getting them on a routine sleep-wake cycle, a routine activity cycle, can help with symptoms quite a lot. And so it seems to be that that it's not just sort of your appetite and cognitive performance and sleep-wake, but like even mood and other like depressive symptoms are linked to clock function. So I guess the takeaway is no matter what, get sunlight in the morning, be awake during the day, and try to get a good rest at night if you can. And it really is, the thing that will unite all of the systems in your body to promote well-being.
Yeah, when I was recovering from a health issue, my acupuncturist said that the best time, like he encouraged me to go to sleep by 10 p.m. and then wake up by 6 a.m. And apparently like organ systems in the body, like you mentioned earlier, repair themselves and heal during the cycles that happen at night.
Yeah, I mean, this is really stunning, April, because we do know at night, we often feel like as humans, we can control our environment, control what we choose to do. We turn the lights on, we stay up, we watch movies, we watch Netflix. And all of those activities, including snacking at night, have really profound consequences because we're really supposed to be in a restful mode at night because our bodies are kind of building up and repairing from the day.
Yeah. Thank you. There's a question from Dominic. It's about your lab. So though I'm sure it changes around, what does an average day in your lab look like?
Oh, that's a good question. It does depend what you're going to do. So we purify a lot of proteins, which means that we actually grow them in E. coli and bacteria, kind of trick them, we coerce them to make our human proteins. So a day might be involved in coming in, taking liters of bacteria food, really stinky food, and growing bacteria up over the course of the day and adding, incurring our proteins to be made. And then purifying those proteins by harvesting the bacteria, smashing them open with a big instrument, and putting them on a series of instruments throughout the day. And so it's a lot like cooking. My brother's a chef, and I really feel like we cook with proteins in the lab. He cooks the food. Now we don't eat our proteins, but we do the same thing. We follow recipes, we grow them, we break them open, we cook them, and we basically spend a full day on our on our feet, moving liquid around and chasing proteins which are invisible to the eye. I like to joke we move around salty water. Little tubes of clear liquid hold all these proteins, and it isn't until we crystallize them that we can actually see them in real life. So it's a lot of hard work before you get that home run of seeing your protein crystallized or collecting data at the synchrotron.
Oh, that's amazing, and I want to thank you again for all the work you do with undergraduates, especially introducing them to the laboratory methods and it's the longer process of deciding what experiments to do next. Right. We have a question from Katie. This might be more of a psychology question or a politics question, but if morning people are in the minority, why do they dictate when we go to work and school?
That's a wonderful question. I think we should open the discussion and talk about all of that. Why is it? Is it because Ben Franklin said the early bird gets the worm? Right? I don't know. It's a great question. I think personally, I would say in my lab, we're really aware of circadian timing, and we do not enforce, a 9 a.m. start or something. People come in when they want. I have a gal who's a total night owl, and she comes in, 10 or 11 and works later into the evening with something that fits her schedule. So I was hoping that with the advent of more work from home with COVID, that maybe people would be able to follow their natural schedules a bit more. But I think fundamentally, my answer to that question is that all is like the political finance system, right? Stock markets have to open at a certain time. Therefore, banks have to open. Therefore, everything is kind of set by global interests. But it'd be great if they were pushed back a bit.
Yeah, I would have appreciated that too in the past. There's a question from Barbie, and this is about one of the research concepts that you presented. Did the Tau mouse have a shorter lifespan?
That's a great question. So, that work, I think is still a little bit unclear, and it's kind of for some details of the genetic work, including like the specific mouse background that the mutation was found in. We do know that if you align your clock and your eating with the day-night cycle that's present, it does extend lifespan. So my postdoc advisor, Joe Takahashi, just published a paper. Again, it was only on male mice from one genetic background. So take that with whatever grain of salt you will. But it was an incredibly in-depth study that went on for 5 years to study the effects of restricting calories or restricting when you eat or both on lifespan. And the results were stunning that we've known for years in studies ranging from like yeast, really simple organisms, up to fruit flies, that if you restrict calories, that the organisms live longer. They're not necessarily healthy, but they live longer, And I think none of us want to live to be 130 and frail for the last 50 years of that, right? We want to be healthy and live longer, or stay healthier longer. And so Joe's work showed really elegantly that if you time when you eat, much like the study I showed you before, that animals stay healthier longer. And not only that, they extend their lifespan by about 30% when you add both like dietary restriction and this kind of timed eating. So for any folks who study intermittent fasting or time restricted eating, there's real science behind that. And that says, because it limits kind of when you eat to your active phase. And so, take what you will, it was male mice from one type of genetic background, but it was the first really comprehensive controlled study showing that it can extend your lifespan if your clock resonates with the environment. So that's my really long-winded way of saying, if you have a 20-hour clock that never matches up, you're probably going to have effects on lifespan. Just no one's really done the experiment in the right way yet.
Yeah, so I have a question from Professor Karplus, and I'll say I actually took his VLSI circuits class in about 1990, and he had a big impact on my career personally. But that's not why I'm asking his question next. So he has a couple of questions here that are kind of linked. He points out that you actually need to add a none of the above option for your chronotype poll that you gave earlier, which I take to mean that Professor Karplus is none of the above, doesn't have a regular cycle. He says that his genome sequence shows a retained intron from Cry1. Could this be an explanation of my rather unusual sleep cycles?
You know, actually, I would love to talk with you offline about this because there was a paper shown again from hamsters. Who would have thought? Syrian hamsters. There was a new allele identified, which they call duper, as in like super duper. You know, I don't know where the name came from, but it was found that there was a change in the intron structure of Cry1 that was just published in the journal PNAS. So we should link up. I'm actually quite interested in that. Those studies also had quite a short period. Now, he's right. I left off a couple of very well-known circadian phenotypes. One of them is called non-24-hour sleep syndrome, where you basically never really line up with the light-dark cycle. And those folks will cycle throughout the day depending on their internal clock. So let's say your clock is 24 hours and 10 minutes. That means every day you're adding 10 minutes with respect to sunlight. And so at some point in the month, your sleep cycle has shifted with respect to the darkness, and you will be sleepy in the middle of the day, right? So that's really well known. And in fact, there are a few kind of somnolent drugs that will make you sleepy that you can use for that. There are also folks who have circadian, it's not so much the circadian side, but the other side of sleep. The homeostatic drive, where they only need a few hours of sleep a night, natural short sleepers. So these folks are like literally superhuman. They sleep maybe 4 hours a day. They tend to have really high functioning multitasking jobs like CEOs, they're positive folks. And I know a few people at UCSF who are studying human short sleepers to try and identify, for example, how the length or time, our need for sleep is linked to the clock. So yeah, there are folks who don't fall neatly into those three things. But you should email me, Dr. Karplus. I would love that.
That's so awesome. There's a question from Lisa that's kind of out of order, but I wanted to ask it now because you sort of mentioned this just previously. Could we adjust to different lengths of day? If so, how much? An 18-hour day, 12, 26, 36?
Yeah, so when we're playing with cells in the lab, absolutely. So it's been shown in almost all organisms, from a single cell cyanobacteria to human cells that we can grow in a dish that have a clock. We can give them specific inhibitors, say for the kinase I mentioned, CK1 delta, and we can make the clock go as long as 30, 40, even 50 hours. But it turns out that individual cells kind of have a pushover clock. They're easy to move around, but in our body, that master clock in our brain is really rigid, and it gets light input and lines up to that very tightly. So it turns out that we can really only adjust to about a 1 hour difference per day. And you know, I mean, the way I'd put it is, you know kind of what daylight savings time feels like for you, right? Like a 1 hour shift in the clock, you feel pretty bad the next day, right? And imagine the next day you did another 1 hour and you had to catch up with that. It's really, it's really hard. And the thing I'll say is that this is like, NASA is very interested in this because Mars has a soul, or a day, if you will. It's a little bit longer than Earth. It's almost 25 hours. And so they have spent a lot of time and resources trying to see if we can use lighting to shift our clock an hour every day so that mission control for a Mars mission here on Earth could live a 25-hour day like they are on Mars and be in lockstep with them. And I'll tell you that only about 30% of people are sensitive enough to light to stay fully caught up to a 1 hour shift day after day. Now, notably, this Cry1 Nidal allele that makes you, where your clock run longer, makes you a perfect resident of Mars. You know, for those folks who have a long clock, you really, it would keep time beautifully with Mars. And so, you know, that's one of the reasons that we're pushing forward, trying to understand the molecular basis of clock protein interactions. Is if we can drug them, we could maybe find a way to, you know, pharmacologically tune or control the timing of the clock, not just for Earth, but for leaving the Earth, you know, space station, exploration class missions and beyond. Fundamentally, we are Earthlings. You know, everything in our biology is linked to a 24-hour cycle that we're not going to have when we go to a new planet. It's very unlikely, right? But you can see this list of behavior and physiology that's so linked to that 24-hour cycle of light. So we do need to work on it.
Well, thank you. I have a question from Joe. And this reminds me, I think there was another article recently in the New York Times I saw that was talking about melatonin and how we're all taking melatonin incorrectly. So maybe you can talk about that as well, like how to, but say, Joe asks, can you adjust or override the clock with drugs like melatonin to compensate for jet lag?
Yeah, so melatonin, I mean, I think one of the issues, and this is definitely veering out of my direct expertise, but my knowledge is that the doses we typically take, your 5 milligrams, 10 milligrams, are vastly over the concentrations that our bodies make. And so often, if you take too much melatonin too late, people get what's called like a melatonin hangover. They kind of feel fuzzy in the morning because they've taken way too much of it. And so the ideal, way we think of melatonin coming on is that it's the synthesis of it is suppressed by light. And as soon as lights dim and go off, our bodies start making it at night, and it promotes sleepiness. So for example, if you were a night owl and you want to try to go to bed earlier, maybe taking melatonin around 8 or 9 p.m. with dim lights would help sort of prepare you to go to sleep a bit earlier. You can do the same thing if you fly, to say Rome, and it's daytime. You could try and when it's nighttime in Rome, but daytime in your body, you could try to use melatonin to make you sleepy a little bit. But fundamentally, that melatonin just makes you a little bit sleepy. It doesn't change the molecular clock that's lying underneath all of that. It doesn't interfere with the gears of that clock. And so there's a whole new group of researchers, both in the States and abroad, who are trying to find molecular targets in the clock to basically make a jet lag pill that would get your clock caught up. And there's been some really exciting advances that have cut down the time it takes to adjust to an 8-hour shift in light, sort of like almost flying to Rome from San Francisco, that cut it down from about a week to 3 days in mice. So I think there's some really exciting things on the horizon. And yeah, so fundamentally melatonin makes you sleepy, but these other clock targeting drugs would get everything caught up in your body on local time.
All right. So there are two sort of related questions, one from Barbie and one from Roxanne. So Roxanne asks, how do night owls survive in a world that celebrates and expects morning larks? And then Roxanne asks, how does a person go from being a night owl to an early lark? It seems to be a difficult transition.
Yeah, so okay, I'll start with the latter one first. And I think, of course, as humans, we can exert control over our physiology like we do in many ways. We could set alarm clocks and force ourselves out of bed, and try to make ourselves go to bed. I think that one's harder. That's if they're talking about the slow decline and change in chronotype as we age, that process, remember, happens over decades in your life, where you go from your latest chronotype in your early 20s, and you get earlier and earlier kind of as the years go. So that particular process, I bet you almost don't even notice, because it happens as you age really gradually. It might take 3 decades to move a significant amount from being a night owl to a morning lark. But we definitely, you can make yourself get up. Sorry, what was the first question was about?
How do night owls survive in a world that celebrates and expects morning larks?
Yeah, it's interesting because I've given this talk before, and I've had some folks who are night owls who loved that fact about themselves. They loved the fact that they would go for runs. I had a friend in my postdoc lab who would go for a jog and write a grant after midnight, well after I was already asleep. And for him, that was prime time. He was awake, alert. It was his time in the day where nobody was bugging him, and he loved it. So, for him, being a scientist worked because he could come into lab at like 11 or noon and work until 9 p.m. and it was fine. I would say that anecdotally, in families that have been studied where delayed sleep phase syndrome or being a night owl is inherited, there seems to be a trend of folks who take on shift work preferentially because they typically, they feel better at night, so they like to work at night. So one way I think some folks have adapted is to move to jobs that maybe require you to work later in the day. And for those unlucky souls who have to get up at 7 and be at work, it's rough. I think the sleep record of that, Cry 1 Delta 11 subject tells the story. Infrequent sleep, napping during the day, it's a rough life. I feel for them.
Question from Jenna. I think she's mentioned that there's a lot of different clock systems in our body, and she asks, can different clock systems within one body ever get out of sync with each other?
Oh, yeah, that's a fantastic question. And yeah, the answer is yes. So I talked about light as a major cue that synchronizes our master clock in the brain to the outside world. But it turns out that all those clocks in the rest of our body are sensitive to other cues. So for example, when you eat and your body makes insulin to tell your body what to do with that glucose, insulin itself is a cue that tells nearby cells, we've just eaten, it must be our active time. And that works as a molecular cue as well to tell your body what to do. So for example, when you fly to Rome, that's my favorite city, if you can't tell, it's my point of reference. When you fly to Rome, and the advice is not just to live on local time, but to eat on local time. Because when you eat on local time, it provides those local and training food cues to your liver and other organs to help speed up their adjustment. So they're not just waiting for signals from the clock in your brain. So by that token, it is possible to have sort of desynchrony within your body. If you think of like the master clock in your brain as the conductor of an orchestra, the rest of your cells and tissues are like each individual instrument. And ideally, they're all working together. But with jet lag, they can get out of sync with one another. And that's generally why you have so many different effects with jet lag, like GI issues, not just sleep, but other kind of problems with your body. So it's a definite possibility to get out of whack in that way.
Got it. And there's this sort of a related question from Wendy, because you mentioned insulin. Does lacking all the hormones affect sleeping pattern? If it is not all, which hormones would affect the sleeping pattern?
So again, that's the part that's not as well studied because again, one thing that, we mostly do work with proteins in test tubes, not with animals. But it's striking because not everybody studies the same processes in both female and male mice. And so sex is a biological variable. It's still not really very well understood. We know that there are effects specifically with some female hormones that affect circadian rhythms and breeding cycles and mating cycles that aren't at play in male mice, of course. They're really just not well studied. And so we know that there are some hormones that affect timing. The things that affect synchronization most are changes in body temperature and hormones like corticosteroids and things like cortisol spikes during the morning to help you like get energized and wake up. So those kind of things tend to work really powerfully to synchronize the clock, and things like other kind of hormones play a smaller role, but still we need more study, I think, to understand the sort of gender specific or sex specific effects of those hormones on rhythms. So I don't have a great answer for you there.
We have a I'll point out we've only got about maybe 5 more minutes of questions, so we'll try to get as many of you as we can. But thanks for all the amazing questions, everyone. Question from Jane. Those of us who have SAD or seasonal affective disorder really suffer during standard time during the winter. Besides using artificial light, do you have any advice?
Well, I mean, that's really the best advice. Artificial light, in particular light in the morning, that seems to be the most important. And I think to me, that's, the hard thing is that the more light we have in the morning, the better our bodies get the daytime cues. And so it's really, all we have right now. Again, if we can find those therapeutics that help to tune up the clock, then that might eventually be something that could help folks with seasonal affective disorder.
There's a question about life at the poles, and you sort of touched on this earlier. So how does life at the poles affect people's rhythms? During the summer, days are almost 24 hours long, and in winter, the night is almost that long. How does a body adjust in those conditions?
That's such a good question. So there are people actively working on this now. I have not seen human studies, but I have seen studies on Arctic reindeer, for example. And this actually was published kind of near Christmas time. So it was almost like unclear if it was a joke, but it was not, most definitely not a joke. And they studied the effects of constant darkness or constant light. And it turns out these reindeer have a way to shut the clock off when they're not getting regular cues. And so, and again, we don't know how that happens. So, we know that there are so many animals who live in really unique environments that still harbor clocks. So for example, subterranean moles that barely see any light, that don't even have eyes, they still have clocks. Cavefish that are buried deep under the earth in ocean and lakes, they don't have eyes, but they still have clocks. So, even if they don't match up with exactly with the day-night cycle, we think they still serve a fundamental purpose and kind of synchronizing all of the activities of the body into rhythms that help to promote systemic well-being. So for now, we know that there are some very interesting adaptations that reindeer do when they hit long days or no days, but the mechanism by which that happens still remains to be seen. I doubt that people living in the Arctic have lived there long enough to adapt in that way. So it just must be very brutal for people. I mean, you can try your best to make light dark cycles by giving yourself 12 hours of light and protecting yourself from light at night to really promote that kind of rhythm. So blackout shades, sister, right?
I have some similar questions from Jennifer and David asking about blue light and specifically. So how does blue light from computers affect our clocks? And do you tape over the blue light emitters when you go to sleep?
Yeah, absolutely. Yeah, I mean, all of us, I think, travel around with rolls of duct tape to tape together towels in hotels, the curtains, and to block out, bright lights. So it turns out this was discovered about 20 years ago. That we all think of our eyes as really being only used for vision, right? And that sees typically a full color spectrum. But it turns out that in the very front part of your eye, the first part of your retina that sees light, which are the retinal ganglion cells, this is the nerves that send signals directly back into your brain. They have a special kind of photoreceptor called melanopsin, an ancient photoreceptor that doesn't send visual cues, but counts photons. It basically counts how much light is out there. And again, this one first makes a pit stop at the clock in your brain and then your mood centers. And the wavelength of light that's best for that ancient photoreceptor, the kind of clock light detector, is blue light. So the reason people put blue light filters on is to reduce that wavelength of light that's really directly signaling just to your clock. I have to tell you, however, all wavelengths of light that you see get sent to your clock. So you can reduce a lot of it with blue light, but red, yellow, green, all of those are still going to signal. So it's a good idea, if you're working late at night on your screen, to have a blue light filter to take some of it out, but it won't be a catch-all because your visual system will also send that information to your clock as well.
Okay, well, I'm afraid we're going to have to stop there. We still have 13 questions in the box. I wish we could get to them all. I want to thank everybody for your amazing questions. And thank you, Professor Partch, for the wonderful lecture.
Yeah, thank you so much. It was super fun.
Yeah, so my name again is Mike Reapy, along with April and our colleague David and fellow volunteer. We're all volunteer organizers. We're alumni like everyone in the audience here. And we do this because we enjoy it. So please join me in a round of applause. You can type it in the Q&A box, but help us join us in thanking Professor Carrie Partch. So thank you, Carrie, for sharing your amazing research with us. And I'll point out that, again, that this talk has been recorded. It will be available on the UC Santa Cruz Arts and Lectures YouTube channel in the next day or two. And we'll post out links to that in our social media and in our thank you emails to everyone who registered. So encourage you, that was fairly technical. Maybe you want to go back and watch it a second time. I'd also like to thank the staff of the Alumni Relations and University Events offices who organized this webinar with us. Specifically, thanks to Shana, Diana, Paulina, and Kristen for providing all the amazing support this evening. Our next Slugs and Steins talk will be Monday evening, August 8th. We actually have not finalized the speaker for that yet, but as soon as we do, we'll be announcing that shortly. We do have a talk scheduled for Monday, September 12th. We will be featuring Yi Zhang from the Computer Science and Engineering Department. She will be lecturing on artificial intelligence and machine learning. Dr. Zhang is also a co-founder of Rulai, an award-winning conversational AI computing platform, which appeared on the Forbes Top 50 Most Promising AI Companies list a few years ago. I'll also point out on July 27th, if you're interested in these lectures, you'll probably be interested in a lecture it's taking place in person at the Seymour Center from 5 to 6:30 p.m. on July 27th. It's called Cancer in the Crosshairs, and it highlights professors Olena Vasky, Michael Stone, and undergraduate student Sachin Parasin. The event will also be live streamed for those of you who don't live in Santa Cruz and you prefer to view virtually. So for more information on that and many other events, you can go to events.ucsc.edu. So on behalf of the UC Santa Cruz Alumni Association, thank you for joining us this evening, and please come back on July 11th at 6:30 p.m. Oh, that's a typo. Hopefully join us on August 8th, 6:30 p.m. for our next virtual event. Good night, everybody.
Good night.